Before meiosis occurs, DNA needs to replicate. DNA starts here, with a nuclear membrane formed around it, and from here, the DNA doubles, so that the DNA has a diploid number of chromosomes. This needs to occur so that a meiosis can occur. And from here, our spindle fibers form from our centrosomes. And within the cell, the chromosomes have now formed their homologous pairs, of which they're attached to their centromere.
The next couple steps are going to be crossing over and the dissolving of the nuclear membrane. For crossing over, the homologous chromosomes are going to form bivalents. These bivalents are going to contain parts of the maternal and paternal genes. We can see here in the colors that there's part blue and part red in each of the bivalents. This is important for genetic diversity because each bivalent is going to contain some of each parent. Eventually, once these split, the haploids are going to contain parts of each maternal and paternal genes themselves as well. After this, the nuclear membrane is going to begin dissolving, and the microtubules coming from the centromeres are going to extend. Once that nuclear membrane is dissolved, the microtubules are going to be able to attach to them.
After the nuclear membrane dissolves, the spindle fibers coming from the centrosomes attach to the centromeres of the chromosomes. They are then pulled to the center of the cell so that the wrong pairs of chromosomes don't end up on different sides of the cell. Then the spindle fibers pull them apart so that the resulting daughter cells will have equal numbers of homologous chromosomes.
After the chromosomes are pulled to opposite ends of the cell, new nuclear membranes begin to form around each set of chromosomes. After this, the cytoplasm splits so that two new daughter cells will form, pictured here.
Here we can see the two daughter cells that are a result of the cell division that we saw earlier of the diploid. The nuclear membrane of both of the daughter cells is going to dissolve, and that's going to allow the microtubules to attach to these chromosomes. Once those microtubules are attached, they're going to pull these chromosomes to the center of the cell. It's important for those chromosomes to be in the center of the cell because we want equal amounts of DNA to be in each of the four haploids that are going to result after these two daughter cells are split.
After the spindle fibers attach to the centromeres of the chromosomes, the sister chromatids are pulled to opposite poles of the cell. This is where we see our second occurrence of genetic diversity because the resulting chromosomes go into whichever gamete is random. Each gamete has the potential to have a completely unique set of DNA. After this, the nuclear membranes begin to form around the four pairs of chromosomes. At the same time, the cytoplasm begins to pinch, and this is important because this will be the framework for the four new granddaughter cells. After the cytoplasm divides, there will be four new
granddaughter haploid cells. This is important because
these haploid cells are gametes that will be used in sexual reproduction.